Broadcast is now starting. All attendees are in listen-only mode.
Hi, everyone. I'm Geraldine Bliss. I serve on the board of directors of the Phelan-McDermid Syndrome Foundation, and we're excited to bring you this webinar today. We will have an update on clinical trials from Dr. Alex Kolevzon at the Stevers Autism Center at Mount Sinai in New York. Before we begin, I am very pleased to introduce you to our new executive director, Ronnie Blumenthal. Ronnie began as our executive director about a month ago, and I think today is her first opportunity to meet you all live and in person. So, Ronnie, I am going to hand it over to you if I can find you on this list here.
Thank you, Geraldine. Hi, everybody, and thank you for joining us today. As most of you probably know, this webinar is taking place on Rare Disease Day, a great day international in scope of recognition for the rare disease community, and that it is in its 10th year, the 9th year that our country has participated. And today, people and organizations all over the world are holding events to raise awareness of the impact of rare diseases, which cumulatively impact 1 out of 20 people. In our community, I just wanted to mention a few people. Our foundation's advocate, Ashley Shaddles, is in DC today and was there yesterday advocating for our families. Our regional rep, Christina Fisher, held an event in Houston at the Health Museum. Janet Mitchell-Wilson is working hard in Oklahoma on a large event, which will actually take place in April. And our friends in England, Victoria Fedderson and Alison Turner, are off to London for a World Rare Disease Day conference at Westminster. And Daniel Wall has shared a few events taking place in Massachusetts, and Nadia Hanim Abdul Latif has been organizing a series of events and PSAs in Malaysia. It's exactly these grassroots efforts that built our foundation and took it to where it is today. And now we are really delighted to be hosting Alex.
Great, thank you. So Alex, I'm going to make you the presenter now. And I will quickly introduce you. Alex, Dr. Kolevzon, as many of you know, has been...
Alex is fine.
Dr. Alex has been working on Phelan-McDermid syndrome for many years now. He's had several studies, including a couple of natural history studies and 3 clinical trials. Is that right? 3 clinical trials now? And today he's going to provide us with a little update on what's going on with those clinical trials.
So thanks, Geraldine, and thanks, Ronnie. It's really nice to be here. I'm excited about being able to share some updates with the community. As Geraldine said, we've been working on Phelan-McDermid syndrome for about 10 years now, both in terms of characterizing patients, but also our main priority is really trying to develop new treatments. So I thought I'd give you an update. And before I do, let's see. Before I do, of course, disclosures are really important. I get support from a variety of sources, foundations, the NIH, and also consult to a couple of different pharmaceutical companies. And importantly, Mount Sinai and Dr. Buxbaum, who's the director of the Stevers Autism Center, have a shared patent for IGF-1 in Phelan-McDermid syndrome specifically. And I'll be talking about IGF-1 today, so that particular conflict is relevant. So as you guys, I'm sure all know at this point, many different genetic causes of neurodevelopmental disorders broadly seem to converge on some common pathways, such that autism and neurodevelopmental disorders are starting to be seen in part as what's called a synaptopathy. So this is a slide that illustrates a glutamate synapse. So the sort of top part of the slide shows what's called the presynapse, and then the bottom part is the postsynapse. And these are examples of glutamate receptors. And glutamates are really critical chemical for learning and memory. And what you see is, maybe you can see my pointer. Can you see my pointer, Geraldine?
Yes. Yeah.
Oh, cool. So what you see here is of course, SHANK3, our favorite protein. And this is a critical scaffolding protein that's anchoring these really important glutamate receptors. And when this protein, when there's loss of function on one of the alleles of chromosome 22, what you see is essentially a disruption in the integrity of the nerve cell connections, particularly around glutamate synapses. So this ends up being very important, and also important to neurodevelopment broadly. You see a lot of other proteins in this pathway, like P10 that relates to Cowden syndrome, or TSC that relates to tuberous sclerosis, all of which cause neurodevelopmental disorders associated with autism. So as part of, in part out of recognition of how important the synapse was, we formed about 4 years ago now a developmental synaptopathies consortium, of which Phelan-McDermid syndrome plays a really important role, along with tuberous sclerosis, along with P10 syndrome. And the goals of this consortium are to comprehensively characterize Phelan-McDermid syndrome using standard medical, behavioral, and cognitive measures. We're especially focused on trying to track the natural history of the syndrome, where we use repeated longitudinal assessments. So every 6 months to a year, we see people in follow-ups to try to figure out what the course of the illness is, and try to figure out, you know, what are good ideal times to try to intervene, for example, and what families potentially can expect over time. We're really very, very focused with both the help of the NIH, but also with really important support from the Phelan-McDermid Syndrome Foundation on developing biomarkers. So we want to try to figure out ways to measure change, but do it in objective and quantifiable ways. And so we're looking at things like EEGs and imaging to try to figure out how we can better design clinical trials with specific markers that reflect the underlying biology and don't rely as much on just clinicians' impressions or parents' reports. And then, of course, the overarching goal is to become ready to roll out large-scale clinical trials and bring new treatments to bear in Phelan-McDermid syndrome. So with that in mind, the pathway we've been following at the Cerebral Autism Center and really across the country and even the world in neurodevelopment is to try to think about kind of a bottom-up approach where we focus on specific genetic causes of autism and neurodevelopmental disorders. We create different kinds of model systems, whether it's with mice models or rat models, or even now neurons derived from humans and using iPSCs or pluripotent stem cells, it's called. We use those models to really better understand the pathophysiology, and in this case, what's going wrong in terms of the nerve cell connections when SHANK3 when there's loss of function of SHANK3. And then we can test specific medicines in the models, and then if those medicines work at reversing either the behavior of the models or the way that nerve cells connect, for example, then we bring those medicines to clinical trials in kids. And that's the pathway we've been following, and I'll show you sort of 3 examples of that in terms of ongoing work at the CBER Center. So going back to 2008, 2009, Dr. Buxbaum in the Autism Center here created a mouse model of Phelan-McDermid syndrome, and we got very interested in the potential of insulin-like growth factor in trying to reverse some of the deficits we saw in that particular mouse. So insulin-like growth factor, you guys have heard about this. You know, we know that this is an important factor for promoting synaptic plasticity or synaptic growth. We know that it crosses into the brain. It's released by the liver in response from growth hormone secretion from the brain. And there's a lot of potential mechanisms of action, but one such mechanism may be triggering a specific cascade in this kind of postsynaptic density that I showed you before, where it's increasing AKT and mTOR to promote protein synthesis and to promote nerve cell growth. So based on models, based on work in the model system where we saw changes in behavior, specifically motor skills, and changes in nerve cell integrity or electrophysiology, basically, we decided to do a double-blind placebo-controlled trial with IGF-1 in Phelan-McDermid syndrome. This is a trial where for 12 weeks people are on medicine, IGF-1 or placebo, and then for another 12 weeks they're on the opposite condition, and we do it in sort of a random order, and neither the clinician nor the family know in which phase you're on which treatment. Insulin-like growth factor is not a trivial endeavor. This is a subcutaneous injection twice a day. In addition to the injections, we're making sure that families are monitoring glucose levels using finger sticks, and we're dosing it very carefully, but based entirely on the child's weight. In the first 9 patients, we saw pretty significant effects on social withdrawal and restricted behavior. We published these results back in 2014. And because of how encouraged we were by the findings, we did a follow-up study with another 10 children. And this is now an ongoing effort that we have at the CBER Center. I released some preliminary results from the second phase, which was a additional 10 subjects. And what we did here, and this is also online, I think it's probably hosted on the Phelan-McDermid Syndrome Foundation website. That's correct, right?
Oh, I'm sorry, what was that?
The document that we released sort of describing the preliminary results from the second phase of the IGF-1 trial, that's online. It's easily accessible.
Right. We do not post it on our website, but it is available online.
Okay, and I'm happy to share it with anybody because we definitely wrote it in an effort to disseminate. So in this kind of second phase, we looked at the results in a lot of different ways. We looked at only the new data, just the 10 new subjects. We looked at the data in a way that looked at only the first phase of this crossover trial. So basically using a very kind of traditional design, which is called a parallel group design, where you only have kind of one phase of a study where half the group is on drug and half the group is on placebo, and you just compare it in that way. And then we also looked much more broadly at the combined studies. And really the key question was, between baseline and week 12, which was the duration of the study, what was the change in various measures when people were on drug as compared to when they were on placebo? And what we saw in the first study that we published was that there was a significant improvement in social withdrawal. When we did the second study, however, even though there was still a big change in the social withdrawal subscale scores when you were on drug, there was also a big change when you were on placebo. So that difference ended up not being statistically significant, unfortunately. We think that there's a variety of reasons for that, and it was disappointing. But nonetheless, we continue to see this kind of clear trend of improvement, and actually the magnitude of the improvement remained the same. So the challenge for us going forward is, how do we minimize that placebo response, right? We did continue to see the significant improvement in restricted behaviors, and we replicated that finding from the first study pretty clearly. And then importantly, we also looked at a lot of different measures where we saw some signals of change in the first study, but they weren't statistically significant. Some of those emerged as statistically significant in the second set of data, especially when we combined just the phase 1 into this very kind of traditional parallel group design. And their hyperactivity emerged as a finding that we thought was really important and was very consistent with what we saw clinically in the patients that were coming through the trial, where they did seem to get calmer over the course of treatment, and they did seem to kind of be able to sustain attention a little bit longer. And we thought that was an important thing for following up in future studies. In addition, what we've started to discover in Phelan-McDermid syndrome is a pretty specific profile of sensory reactivity, right? In neurodevelopmental disorders and in autism broadly, kids can be very sensory reactive. They can be hyperreactive, hyporeactive, or sensory seeking, and there's a lot of variability. Within Phelan-McDermid syndrome, though, it seems that the most prominent profile is one of hyporeactivity, and that goes along, of course, with the high pain threshold that you see in most of your children, and that also goes along with a kind of a sensory seeking type of behavior, and a lot of the sort of the chewing, for example, could be consistent with that. So in our data set, the new data set, we saw something that was consistent with what we were observing in the clinical trial based on what parents were telling us, is that the sensory profile was sort of changing, in essence, that kids seem to be less under responsive. And what you see in this graph here, like if you look at just the one on the top left, higher scores are actually closer to sort of normal in this case. This is sort of a movement towards a more typical profile, whereas lower scores are greater deviations from a typical profile. And so what we saw was that the under responsiveness seeking sensation domain and the modulation related to body movement, you see me tilting my head, the modulation related to body movement and position, those are sort of two domains that jumped out at us as statistically significant, even though they weren't in the first trial. I don't know exactly how to explain that, but in sum, to us, it feels like something that's really important to follow up on. And of course, this is a really critical domain in terms of day-to-day functioning, because we think that things like high pain thresholds, for example, can really be very problematic when kids aren't describing pain, and when they can lead to things like infection and so on. So again, in addition to being burdensome in terms of administering IGF-1, it's also not trivial as far as the potential risks. So in clinical studies with patients that didn't have Phelan-McDermid syndrome over very long periods of time, over almost 4 years, about 5% or more had side effects of things like hypoglycemia, tissue swelling, bruising, ear infections, a lot of sort of problems related to increased growth, like tonsillar hypertrophy, which is an increased growth of the tonsils, headache, dizziness, even convulsions, some vomiting. Some of the smooth muscle of the heart may have changed. There's cardiac murmurs, joint pain, pain in extremities, and also an increase in the growth of the thymus, which is an immune system organ right behind the sternum. So these are all the things that we were worrying about and that we were monitoring closely. In the 12 weeks within the patients that we saw, the 19 patients with Phelan-McDermid syndrome, and you see that here on the right, we really didn't see any serious adverse events. What we did see, of course, was an increased risk of low blood sugar, although really almost none of the patients had any significant clinical symptoms as a result of this. So even though their blood sugars might dip below what you would expect it to be, they didn't actually develop like seizures or convulsions or anything extremely concerning. We did see tissue swelling in a couple of kids. We did see, of course, bruising at the injection site when you'd give the injection. There were kids who had worsening of constipation, increased appetite in some kids, decreased appetite in others. There were some kids who had worsening of their sleep problems, a couple of kids with irritability, energy changes, and there was, I think, at least 2 kids with increased biting and chewing behavior. So again, these are all, these are significant side effects. They occurred more frequently among kids who took the IGF-1 as compared to placebo, but none of them to us represented any significant danger, and nobody had any what's called serious adverse events.
Alex, what constitutes a serious adverse event?
So a serious adverse event is something that's, of course, life-threatening. It's something that requires hospitalization. It's something that requires like immediate emergent medical intervention.
Okay, thanks.
Yeah, and it's very clearly defined in our consent form, and we have to report it back to our patient safety boards, and we take that stuff very, very seriously, and thankfully we didn't see that at all. And then, in order to be safe, we monitor a whole bunch of things. So every 2 to 4 weeks, people are coming in, they're getting physical exams, neurological exams. We're looking at the back of their eyes to make sure there's no increased pressure in the brain. We're looking at their tonsils. We're monitoring their heart using EKGs and echoes. We're doing bone x-rays before and after each treatment phase to make sure there's no unwanted skeletal growth. Of course, kids, when it's relevant, are getting pregnancy tests. We're looking at height and weight to make sure there's no significant increase in growth. And so we're doing a lot of safety monitoring in this trial. And that's one of the things that makes us comfortable giving the medicine on one hand, but it's also one of the things, of course, that makes it, challenging for families to kind of, adhere to. Everybody that we work with, obviously was able to kind of complete the study with the exception of one patient, but it's not trivial. So that's the IGF-1 story, and that's kind of a brief update where we are. I can say with a lot of gratitude that we are, really excited to continue this IGF-1 program. We just got a very large grant from the NIH to actually continue it and to embed biological markers to do a better job of figuring out in advance who may be likely to respond to IGF-1. We think that's obviously a key. It's clear that not everybody responds to it. Some do, some don't, and the challenge for us is just try to figure out in advance how to potentially stratify groups and figure out in advance who's a likely responder. And we think we have some leads as far as that's concerned based on some of the biological marker development that we've been doing for the last several years. So the next medicine that I wanted to talk about and update folks is oxytocin. So oxytocin is this pro-social hormone. It's a natural hormone. It's released by the brain. It's been seen as a love drug. It's been seen as a trust medicine. People talk about it as sort of a panacea for all kinds of social problems. It's clearly not that, but we do think it may have an important role in autism. And we've just finished a 300-person trial across a 6-site network, specifically studying oxytocin in autism. And all different kind of severity levels of autism, and we're hoping to see some results in the next couple of months, actually. But in the meantime, because of all the interest in oxytocin, Dr. Buxbaum and his group, including Hala Harani-Nicolas, used oxytocin in another model system. So instead of using a mouse model, they created a rat model. Rats have bigger brains; they're a little bit easier to work with, evidently. And they looked at some of the electrophysiological measures. So this is a measure of what's called long-term potentiation. It's basically a proxy of synaptic plasticity. And they looked at the effect of oxytocin on these Scheng-free rats. And they found that actually oxytocin reversed the electrophysiological deficits. And that's a really important marker of nerve cell conduction integrity. And we saw that there was essentially a reversal of the deficit. And that was a very exciting initial finding. Hala also looked at things like social recognition memory. Obviously for us, the social domain is a very important domain. There are lots of different kinds of paradigms that you can use to measure social recognition and things like partner preference in animals. And what Hala and the group saw is that under the influence of oxytocin, deficits in social recognition were rescued with oxytocin. Another really important paradigm, and one of the things that kind of points to some of our new work, which is to try to figure out ways of measuring things in animals that are very similar to ways that we measure things in humans, because I think one of the reasons that clinical trials may not work in the clinic is because they don't totally replicate what's being done on the preclinical side with animals. And lots of times, people have done wonderful jobs, curing all kinds of diseases in animals, and then of course when you kind of move that to the clinic with humans, it fails. And so this is something called the 5-choice serial reaction time task. And for any of you who are familiar with this, the continuous performance task, it's very similar in humans, where they're basically training these animals to tap a light in response to that light kind of lighting up. And so there's these kind of 5 chambers, and when the chamber lights up, the mouse taps, or the rat in this case taps the light, and then it gets a reward. And you train the rat to do this, and rats that are missing a copy of their SHANK3 gene do a terrible job of this. They have a lot of difficulty correctly identifying the light, and then when you don't have a light, they have a lot of difficulty inhibiting the impulse to tap one of the squares, if that makes sense. And so those are called correction and omissions. And actually, under the influence of oxytocin, there were very significant improvements in both the number of lights that were correctly tapped and the number of times that the animal was able to withhold the response. So we thought that was kind of a really important measure of attention and perhaps of learning. And it's something that we can somewhat replicate in clinical trials with humans also. And so this was enough for us to start thinking about doing a clinical trial in kids with Phelan-McDermid syndrome, which we started a couple of years ago, actually maybe 18 months ago. And so this is a much easier medicine to get access to. We're importing this from Europe, where it's actually commercially available. It's an oxytocin nasal spray. Oxytocin used to be made as a nasal spray in the United States. It was since discontinued, but the European Union continues to make it. So what we were able to do is a placebo-controlled trial for 12 weeks, and then everybody after the 12-week phase, where you don't know whether you're on oxytocin or placebo, everybody gets open-label treatment, so active treatment with oxytocin for another 12 weeks. And there are a lot of study procedures as usual. We're looking at many different kinds of outcome measures, both clinician measures, parent report measures, and a variety of objective measures, including things like visual evoked potentials and some new tools that were developed at the Seaver Center that are trying to tap into sensory sensitivity. And so, this is it's kind of a, it's a somewhat burdensome trial in the sense that there are a lot of visits, but we think that the safety profile is extremely reassuring. We are pretty careful with our titration, so people are starting on basically 3 sprays twice a day, and then they get up to 6 sprays twice a day if it's well tolerated, and then we monitor that over the course of 12 weeks. Some of the potential side effects are things like headache, increased heart rate, or decreased heart rate. And I love when that happens with medicines. It can kind of go in both directions. Nausea, vomiting. There's some potential for it to affect smooth muscle of the heart, so that you could have irregular heartbeats. Blood pressure can change. Some kids, in fact, even kids with Phelan-McDermid syndrome, we saw had a little bit of increase in irritability. Some kids gain weight. It can affect sleep, as always. Some kids can be a little bit disinhibited, meaning they have trouble kind of controlling their impulses. So, for example, you're taking oxytocin, it makes you want to engage more socially, but you continue to lack the social skills. Some kids might be much more active in trying to engage, but it's not appropriately, and that can look kind of disinhibited. And there was some evidence of increased activity levels. This is all in a broader autism population, and these side effects are, at the moment anyway, not relevant specifically to Phelan-McDermid syndrome. We're still collecting that data. So far, I think in the 7 or 8 kids that have come through the trial, I'm not even sure it's that many, we really have not seen any significant side effects other than some increase in irritability. And as always, we're monitoring things very closely with physical exams, neurological exams, heart exams. We're looking at vital signs, and we're just sort of gauging side effects based on parent reporting at every visit. So that is the oxytocin story. How much time? How are we doing with time? Okay, so we've got 10 more minutes, and then I think we'll open it up for questions. Yeah?
That's perfect.
Okay, good. So this was very exciting. For the first time in Phelan-McDermid syndrome, a pharmaceutical company actually came to approach us, and I think that that's a really important sign that Phelan-McDermid syndrome is gaining a lot of momentum, that people are starting to understand that the SHANK3 gene is really critical for neurodevelopment, and particularly to understand that if you can develop treatments for specific single gene forms of neurodevelopmental disorders like Phelan-McDermid syndrome, those treatments may have relevance to autism and neurodevelopment more broadly. So this company, Ammo Pharma, is a small pharmaceutical company, came to us with a specific drug called Ammo-01. And we looked a lot of the preclinical data and thought about some of what was seen in the preclinical data and decided that a really important domain to go after was epilepsy. And so Ammo-01 is an inhibitor of the mitogen activated protein kinase pathway. This is a really important pathway, of course, for neuronal survival and synaptic plasticity, as we've been talking about. And basically, it prevents activation of this RAS-MEK-ERK pathway. And so it's essentially an inhibitor. Specifically in Phelan-McDermid syndrome, it was studied by taking a SHANK3 mouse and knocking out both copies of the SHANK3 gene. And what they did was they measured basically the ERK levels specifically. And they also measured a different kind of parallel pathway to see if the effect on ERK was specific. So in the SHANK3 knockout mouse, the RAS-ERK pathway activity was markedly increased in the cortex, and not so with the AKT pathway, which is this kind of parallel pathway. There was an increase in the AKT pathway in the cortex, but not in the hippocampus. But the magnitude of the increase with the RAS-ERK pathway was much, much higher. And so anytime you've got like this significant activation of a specific pathway, you think about, well, maybe that's part of the problem with the underlying biology, and if you can inhibit it or decrease the activation, you can have an effect on the phenotype. They also saw that the RAS-ERK pathway was activated in lymphocytes and blood cells of these SHANK3 mice. So what was done, and this is again, it's following the same pathway. We're doing it, other companies are doing it, obviously. So they took a single dose of this compound, Ammo-01, at 30 milligrams per kilogram. This isn't a mouse, so it's injected into the peritoneum or intraperitoneal. And it was compared to placebo, and this is 10 different mice. And it was clear based on levels that were achieved in the plasma that actually you were getting to places in terms of the level of this compound that were consistent with what was seen in clinical studies in the oncology arena. And this is an important point where Ammo-01 was a drug that was developed initially for cancer to kind of prevent tumor growth, for example. And so what they did, as is typically done, is they looked at many different kinds of behavior of the SHANK3 knockout mouse. And what they saw were things like increased anxiety, excessive grooming, impaired social recognition, like we talked about, sensory motor dysfunction, and sort of deficits in some typical species behavior like marble burrowing. And then, of course, and this is really important here, you can actually stimulate mice with missing copies of the SHANK3 gene, in this case, where you basically ring like a very loud bell and you can induce a seizure. These are called audiogenic seizures. And remarkably, all of these phenotypes were rescued under the influence of this drug, Ammo-01, okay? And so for us, that was like, kind of important sort of proof of concept that there could be some real relevance to some kids with Phelan-McDermid syndrome. So the next thing, obviously, is really making sure that this is a safe drug. There's been a lot of safety data, both in rats, primates, and then I'll talk about briefly in humans. And what seems to be is that the toxicology profile is safe. There are no significant problems in terms of degenerative changes. There's no major effects on other organ systems. In totality, the animal data seem to suggest that you could give up to 14 days of continuous treatment in humans at relatively high doses actually that are 4 times the dose that we're proposing in Phelan-McDermid syndrome. This 120 milligrams is per meter squared, which is a measure of body surface area. Where is my mouse? There it is. And because this is a drug that was developed in cancer, there's already a fair amount of clinical data. So there have been 26 patients, I think another 20 patients. So at least 46 patients that have had cancer that have been given this drug for sessions of, I think, up to 14 days. And as of yet, it seems as if the serious adverse events that occurred were things that are typically common in this kind of patient population, which are obviously very sick folks, and were not deemed related to the MO01, and were also not deemed related to the dose. But there were very serious adverse events that happened, like anaphylaxis, rash, fever, pneumonia, thrombosis, which is like a clotting. There was like a pleural effusion, which is sort of air in the lungs, essentially, diarrhea, shortness of breath, and a bloating of kidneys. So these are all, obviously very serious things. But again, none of them were thought to be related to the drug. And true also in a specific subset of people with a brain cancer, glioblastoma, where they saw fatigue, headaches, muscular weakness, anxiety, seizures even, which is a little bit peculiar, given what we're using this for. And there were very serious adverse events, but only in 5 patients, and again, not related to MO1. And the consensus of all the various folks that have kind of reviewed these data, including the FDA and regulatory bodies, is that clinical safety data in cancer patients suggests that even 14-day cycles of MO has the potential to be safe and well tolerated. So we are very interested in trying to, of course, evaluate the safety and tolerability of this particular drug. We're using it in a single 6-hour intravenous infusion. There is no placebo group. Everybody's going to get active treatment. We're also very, very interested in whether or not this medicine can reduce seizure frequency by at least 25%. And we're going to also use a whole variety of other measures to see if we can see changes over the course of about 4 weeks post intervention in other domains, and all the typical domains of function and just clinical global impression, and of course whether we see change in some of our biomarkers. So for inclusion, we're trying to get studies that are involving adults. We think that obviously this is a really important thing, and to date we've sort of neglected the adult population in terms of clinical trials, and we're also very focused on people with pretty severe histories of seizures. So this is going to require that kids have at least 4 seizures a month, and that it's not just a very significant uptick in their seizures, but that kind of degree of frequency has been occurring for at least 6 months prior to screening. We also obviously want kids to have things like their anticonvulsant regimens to be stable for the last 4 weeks before screening. They can't be sensitive to any of the formulation components of the drug. They can't have uncontrolled hypo or hypertension, so not high or low blood pressure. And they can't be taking, in the last couple weeks, any major blood thinners like Coumadin or heparin. The study is going to have 4 phases. We're going to do screening, just to make sure that people meet criteria and that families understand what's involved and everybody feels comfortable with it. We're also going to get a pre-medication seizure frequency. At baseline, they're going to do assessments in the clinic, and then immediately afterwards, they're going to receive a single 6-hour infusion at a single dose of 120 milligrams per sort of meter squared, which is a body surface area measurement. And then we're going to do follow-ups at 1, 2, and 4 weeks after receiving study medication. And there is the hypothesis anyway that even one single intravenous infusion is going to have longer-term effects. So we do expect to see, or we hope to see anyway, a decrease in the seizure frequency. We're going to be measuring, as usual, lots of different things, but most importantly, there's going to be seizure diaries that families bring home and monitor their children very closely. We'll be monitoring adverse events. We'll be doing biomarkers, sensory sensitivity, clinical global impression, and a lot of safety labs as well. So that is the end, actually, of the section on the clinical trials. So as I said, and as Geraldine said in the beginning, we've got these three clinical trials going on that we're really excited about. We rely heavily on families to participate in order to try to learn more and figure out what hopefully is going to be helpful going forward. The other really kind of exciting thing that I wanted to share with the community is another study that the Phelan-McDermid Syndrome Foundation has funded. It's now the second pilot study that the foundation has funded as part of our rare disease network. And the goal of this study is to characterize the syndrome across the lifespan. So we want to actually capture adults. So we're going to be looking at adults 21 and over, and that's to complement the natural history study that's being funded by the NIH that looks at kids from 3 to 21. This is the same as the natural history study in many ways. It involves 2 to 3 days of testing, including cognitive testing, a lot of caregiver interviews. We're focusing a lot on sort of any history of cognitive changes, any changes in terms of adaptive functioning. We're trying to better understand this subset of people that are actually losing skills. So we really want to start to include more and more adults in our study, and the long-term goal is to capture kind of a big cohort of adults and then continue to monitor them over the time. And this is occurring not just at the Seaver Center, but at the NIMH, at Rush in Chicago, UT Southwestern, Boston Children's Hospital, Stanford. And the really other important thing is that we recognize a lot of these adults have lots of difficulty traveling, and so much of our assessments have been tailored to do either in person or remotely. And if they're being done in person, we'll also have the opportunity to provide comprehensive reports. And these are our study contacts. So Jordana is a research coordinator at the Seaver Center, and she's coordinating the AMO and the IGF-1 study. Kristen is another research coordinator at the Seaver Center. She's coordinating the oxytocin study. And Allison Durkin is another research coordinator at the Seaver Center, and she's been in charge of the RDCRN studies, and she'll be coordinating the adult phenotyping. And for anybody that's interested, you can contact her for the adult phenotyping study, and she will refer you to the closest rare disease network site so that you don't have to travel. We do have travel funding available for the children in the rare disease natural history study. The adult phenotyping study so for people over 21, we unfortunately don't yet have travel funding available. And that is it. Thank you very much for your attention. Really happy to take any questions.
Great, Alex. We have a few questions that have come in, and I have a question also. So you mentioned using biomarkers as an objective outcome measure in clinical trials. So the Developmental Synaptopathies Consortium has a pilot study using the the visual and auditory event-related potentials. Are those the same biomarkers that are being used in the oxytocin trial and in the AMO1 trial?
They are. So biomarkers have lots of different potentials. So one is to try to capture the biology of a given syndrome. But to use that to sort of stratify within the syndrome. So there are perhaps subtypes within Phelan-McDermid syndrome that have certain profiles on visual evoked potentials, and others that have kind of different other types of profiles, and that's important for us to know. But for me in particular, in terms of developing new treatments, one of the most valuable uses of biomarkers is to actually see if you can use it to measure change over time. So if there's a given signature, let's say on a visual evoked potential, and that signature indicates dysregulation or impaired regulation of glutamate functioning, which is what's exactly happening in Phelan-McDermid syndrome, is it possible that by providing a treatment, you can correct some of that glutamatergic dysregulation? And that's what we're hoping to see in the context of our clinical trials, and we already have some preliminary evidence that may be the case, including with IGF-1 and oxytocin, in fact.
Great. So you'll also add it to the IGF-1 clinical trial, the third clinical trial of IGF-1?
Every clinical trial going forward.
Great. Wonderful news. Okay.
Yeah.
Good. Well,
and it's not, we also have different kinds of biological markers. Hopefully we'll be doing some like eye tracking. I mean, there's a lot of, people that have been through realize that we put them through all kinds of tests, and we're really hoping that some of them are going to be extremely informative in the future.
A lot of families may have already participated in your gait study that you did a few years ago. I'm wondering, did anything come out of that in terms of biomarker development? What is the status of that now?
Oh, thank you so much for bringing that up. So we collaborated with Elizabeth Torres as part of the IGF-1 study to do a gait analysis where Elizabeth basically connected these kids up to 16 different electrodes and was able to quantify gait in a very specific way. And I was especially interested in whether gait changed, which we were hoping it would based on some of the animal models, pre and post treatment with IGF-1, and that data is still being analyzed. But what she did find is at baseline, comparing kids with Phelan-McDermid syndrome as compared to other kids with autism or developmental disorders, there was a kind of a specific profile or signature really, where kids with Phelan-McDermid syndrome had a lot more fluctuations in their intentional movements. There was more sort of noise in the movement, which makes some sense given that the SHANK3 gene plays not only a really important role in the brain, but also in sort of muscle connections. And so there seemed to be something unique within Phelan-McDermid syndrome that you didn't see in autism more broadly that could reflect something of a biological marker. And that was published actually. And I'm happy to share that paper.
Okay, great. So we've gotten some good questions here. The first is, when will you publish information to the families that have participated in the clinical trials? And is there funding to continue the trials to the next phase?
So I think that question probably refers to, when will I tell families which phase were they actually on drug? And I would love to tell families which phase they were on drug. I think this, obviously you put so much time and effort into participating. That's like such a key feature. The problem is that if I do that, it compromises the studies going forward. And because we do have funding now to continue the IGF-1 studies, and because we are committed to this program more broadly, it's really not appropriate to essentially break the blind among families until we've really moved it to the next level. The next level would be a phase 3 multi-centered controlled trial. And so in terms of when are we ready to do that, we now have this network that's part of the natural history study. We have the same network that's able to implement some of our biomarkers. And so the next step is getting enough funding to actually do a multi-site trial with IGF-1, for example, across our network. And I think we are still some ways away from that. But we just got a very large grant to do another clinical trial with IGF-1, including our biomarkers, just at the Sever Center. So I think the next step after that would actually have to be a multi-center trial.
Okay, great. What percentage of the kids would you say responded to IGF-1?
Yeah, so I could actually answer that exactly, but I don't know the answer off the top of my head. I would say it was probably about 60%. And I think the more interesting thing is that when people responded, it was very dramatic. So there were a lot of kids that just didn't respond at all. It seemed as if it was no significant change. And then there were a lot of kids where we were like, wow, this is remarkable. It was evident to the parents, it was evident to us, even within a few weeks.
Were you able to relate the change in response to drug to any other of the data that you were collecting, any potential biomarkers, any other indicators in any of the outcome measures?
So it's really very preliminary, and there does seem to be change in some of our biomarkers, yes. The most obvious thing to look at would be looking at just IGF-1 levels, and there didn't seem to be significant change there necessarily, but we have a lot more digging to do.
Okay. Okay.
19 subjects, while it took a long time, it actually is still a very small sample.
Okay. Great question here. How can we get our child into a study? And I think you've just answered that with the study contacts that you've listed.
The slides right there, yeah. So there's lots of studies going on, not just the Sever Center, but many of the different sites that are focused on Phelan-McDermid syndrome. And I think I really encourage everybody to kind of get involved. Obviously, a clinical trial is not for everybody. Those are pretty intensive. But I think at the very least, natural history studies, biomarker studies, parent questionnaires, the patient registry, all these things are so important for us to really move the field forward.
Okay. If your child has a VNS implant, a vagus nerve stimulator, can they still participate in the trials?
So that's a really good question. And I think, so I assume that relates. So the answer is yes. The only question would be whether or not they could participate in the AMO study, and that would be determined on the basis of what their seizure frequency is, despite the implant.
Okay, great. Thank you for studying PMS and your efforts with the clinical trials. What is the next step in the IGF clinical trial? As you mentioned, you're going to be, you've got a third phase of it coming along. Are you enrolling more PMS kids in the IGF clinical trial? And yes, you will be. How many, Alex?
So great question. So we have now, so I'll tell you kind of the goal with the IGF-1 program is twofold. So on one hand, we obviously want to push this forward in Phelan-McDermid syndrome, but we also want to use what we're learning about Phelan-McDermid syndrome and about the kids who respond to IGF-1. We want to use that to inform autism more broadly, meaning idiopathic autism, where we haven't identified a genetic cause. And I think that's going to be important for a variety of reasons, but in particular because that's going to give people much more faith in this notion of tackling single rare genetic causes of autism, because it won't just be about the kids that are only affected with Phelan-McDermid syndrome, but about the whole universe of people with developmental disabilities. So we have now 2 grants that will allow us to recruit about 45 more kids with and use IGF-1.
Okay.
So we have funding.
The next question this parent asks is, what are the criteria for becoming enrolled in for participating in the IGF-1 clinical trial?
So the IGF-1 clinical trial, currently the criteria are really that you need to be pre-pubertal between the ages of 5 and 12, or really 2 and 12, which is what we're about to change. So that's not official yet. That's not approved by IRB, which is important. What's currently approved is 5 to 12. You know, there's a lot of different medical assessments that we have to undergo, and there's some clinical assessments, so you have to have certain severity on some clinical instruments. But I think really the vast majority of kids with Phelan-McDermid syndrome will be eligible, assuming that they're between these 2 ages. And you know, you just have to come in and sort of meet us and undergo an assessment, and then we would figure it out together.
You had mentioned the inclusion of the visual and auditory event-related potentials, the biomarkers in the IGF-1 trial. Is there going to be any other significant change to the protocol for the IGF-1 trial?
Compared to the previous ones?
Yes.
Some more measures. We're adding some measures and we're dropping other measures, in essence.
Okay.
I would say. I think a lot of the measures that we know are not going to change. We've just now eliminated because they're just overly burdensome.
Okay.
The age range has changed a little bit, but otherwise it's generally the same design. In an ideal world, I wouldn't do a crossover study. I wouldn't have these 2 phases like this. I would actually just have a parallel group design with an open label extension. And we might be able to do that, but it would depend on getting the drug for free, which we're also working on with the company, but we haven't been successful yet.
Okay. Oh, this is an interesting question. If my child participated, if my child participated in one of the first IGF-1 trials, can they rejoin the 3rd trial?
So, you know, it's a great question. The answer is probably not, unfortunately.
Okay.
Yeah, because then what you bias results, because the people that would want to participate in the 2nd trial are the ones that felt that they responded to the 1st trial, and then you're really not. It's not random anymore. It's not a randomized control trial.
Okay, so we've gotten 2 questions that are sort of related to a different idea. They're both related. One is how can CRISPR be used to help PMS? And the other question is also related to gene therapy to correct a SHANK3 mutation and reintroduce, or to create a repair for the mutation.
Yeah, so gene therapy, you know, we are obviously very, very interested in, and I think we've just been very fortunate to receive a big gift from the CRISPR Foundation, a strategic gift that is in part going to be dedicated to really improving our efforts around developing new treatments, including gene therapies. And in terms of CRISPR, you know, I think CRISPR is really relevant on the sort of model system preclinical side. I am more of a human person. I mean, I am a human person, but I deal mostly with humans. So I think that in terms of testing compounds on the preclinical side, CRISPR is extremely valuable in terms of, you know, manipulating different models and creating ways of actually testing drugs.
Okay. In the oxytocin trial, do you measure success at I think it's the right learning challenge in human subjects? Let me make sure I've read that right. In the oxytocin trial, do you measure success at I think it's right learning challenges in human subjects? I guess maybe the question is, are we really using the appropriate outcome measures?
Oh, yeah. I was going to try to extrapolate that question from the question. Well, so we are trying to tie our clinical outcome measures to our preclinical ones. So we are looking at measures of attention using things like eye tracking. We are looking at measures of electrophysiology using things like visual evoked potentials. And then we're kind of blanketing all the different kind of behavioral domains. So repetitive behaviors, sensory functioning, autism kinds of symptoms, levels of activity. We are really trying to be very broad. I don't think that we're at a stage where we can say, oh, everything's got to be focused on this particular domain, because we simply don't know yet. I can tell you, again, broadly, the idea behind oxytocin is that it's a pro-social hormone. So the idea is really that it's going to be increasing social motivation, or perhaps decreasing social anxiety, or maybe even like increasing the salience of social cues. So obviously the social domain is very important to us. Whether or not that's the right domain, we don't obviously know, but I think that's part of the reason that we do these trials, is to try to figure it out. And then going forward, we would design a trial more specifically based on what we see in the pilot trials. That's the sense of the pilot trial.
I think outcome measures that I think almost all parents would say are extremely important are improving learning and improving communication, especially speech. Are there measures that include both of those, or either of those?
So we do have measures of language, obviously, and indirectly we have some measures of learning. The challenge there is one where 12 weeks is kind of the limit of our trial, and 12 weeks probably is not enough time to really see robust changes in language or in fundamental aspects of learning. I think, if we were to see signal and some evidence of change in the course of a 12-week trial, then you do a 6-month trial, or perhaps a 12-month trial. I mean, the way that this trial is structured, the oxytocin trial, is that you've got 12 weeks of drug and placebo, and then 12 weeks of active drug, which means that about half the patients will have 6 months of drug. And it may be that 6 months is enough to see changes in cognitive abilities. And if we do, then of course the next follow-up trial would incorporate that and would make sure that it was long enough to really detect the change. Mm-hmm. But there are sort of limits in terms of feasibility. And I don't want people to think that if you don't see change in language, for example, the drug isn't going to work on language. But I also don't want people to expect that there should be changes in language over the course of 12 weeks.
This is an interesting question I don't think I've ever heard posed before. Has there been success using neurofeedback?
Using neurofeedback?
Mm-hmm.
Not that I'm aware of. I think neurofeedback or biofeedback in general often requires a pretty high level of cognitive functioning. So you have to be able to kind of follow directions and go through different paradigms that I'm not sure are going to be entirely relevant to as many kids as we'd like them to be in Phelan-McDermid syndrome.
Thank you for your work in studying PMS. Have there been any considerations to using some of the new blood glucose monitoring systems that do not require a finger stick for the IGF-1 study? Now, that's a really important question for families maybe whose kids have lymphedema or other conditions where maybe you don't want to stick them that much. Have you explored?
Our protocol does not restrict us to any particular strategy of glucose monitoring. As long as we get levels, and as long as we feel comfortable having the drug administered after the levels is drawn, I'm happy to consider any types. So people should be sending me things that they want me to explore by all means. Mm-hmm. But right now, our systems are free, so we just need to keep that in mind. I mean, the companies will hand out these glucose monitoring kits for free. The only costs associated with it are buying the test strips that are nominally expensive. But if we can come up with an inexpensive way to measure in a more benign fashion, I'm happy to do it.
Okay, great. So here's a question relating to a couple of questions ago. Has the IGF-1, has there been any increased speech for the PMS kids that are nonverbal? Have you noticed that?
Speech, no. And in terms of the language measures that we use, we did not see meaningful changes on the language measures. I think that the things that we saw in terms of adaptive functioning were more related to kind of motor functioning, attention, level of activity, sensory reactivity. I mean, I think that there may have been some kids where the parents described more functional communication and even perhaps some words. But in terms of the broader group, we measure that with a couple of different things, and we didn't see significant differences.
Okay. a question here. Maybe you want to tackle this one too. What is the difference between a SHANK3 variant and a deletion? And a follow-up question, what is the relationship between deletion size and level of phenotype severity?
Okay. So SHANK3 variant usually refers to a point mutation, and a deletion means that the whole section of often the end of the 22nd chromosome is knocked out, whereas different variations can be just a single base pair, where there's a variation in the sequence of the base pairs, and it can lead to what's called a premature stop, for example, and then you don't code for the protein, so there's still loss of function for the protein. But in the cases of mutations or variants, the only gene that's affected is SHANK3, and in the case of deletions, it could be that a handful of genes are affected to many genes affected, depending on the size of the deletion. And this issue of the relationship between the type of mutation or deletion, or the genotype, as compared to the phenotype or the clinical characteristics, is a really, really important question. And I think that there does seem to be a lot of emerging evidence that often kids with much smaller deletions have less medical comorbidity, may have more intact motor skills, have perhaps more chances of being verbal. So I think that overall, the sense is that the severity has some correlation to the size of the deletion, but it's absolutely not perfect. I see kids with large deletions who are doing very, very well, and I see kids with very small deletions who are not doing well. I think that there will be a relationship, but there's a lot more to learn, because there are going to be other areas across the whole genome that can act on the SHANK3 region to either perhaps compensate or maybe to exacerbate the deficiency in SHANK3. Other things that seem to be emerging from larger patient data sets, and by the way, that's the really key component here. We need to be able to look at probably thousands of patient samples in order to draw reliable conclusions about the relationship between the genotype and the phenotype. But one thing that's, I think, interesting and important is that with very, very small deletions, even deletions that only, or even variants or mutations that affect only SHANK3, that's probably that is sufficient to cause things like epilepsy. That is sufficient to cause intellectual disability. It's sufficient to cause language delay. So much of the phenotype is still going to be boiled down to just the SHANK3 gene, whereas much larger deletions, you might see a greater likelihood of things like kidney problems or heart problems, for example.
And I'll just offer, that one of the ways that we can try to understand how genotype relates to phenotype is through our patient registry, the Phelan-McDermid Syndrome International Registry. So for those of you who are listening in, if you have not yet participated in the PMSIR and the registry, we'll post a link in Facebook, and please be sure to sign up or at least learn more about it and learn how you can participate and help answer that question. How does genotype relate to phenotype? And I know at Sever Autism Center, you guys have some other studies, not the studies that you've discussed today, but some other studies that are looking at sort of the role of sort of sequence variants, not just in the 22q13 range, but outside of that range. You're looking at how sort of spelling errors or differences in spelling of different regulatory regions might alter the expression of critical genes for the phenotypes of relevance in Phelan-McDermid syndrome. Do you want to add anything to that right now? Or
no, just that we have a database now of, I think, over 100 biological specimens from patients. And there's so many amazing things that you can do with technology now. And so, yeah, we're very, very interested in how other regions or other kind of expression profiles play into the severity of the phenotype of Phelan-McDermid syndrome. And as a clinician, to me, there's nothing. Well, among the most valuable things is trying to be able to predict what the course of illness is and what the critical points of trying to intervene are. And that's something that you can hopefully eventually learn more about by studying exactly which kinds of variations are occurring and what else across the genome is impacting the phenotype.
And at our family conference at 2016, your colleague Joe Buxbaum talked about sort of what we kind of referred to as a genetic superheroes project or a resilience project, where potentially there are genes that may be protective and may prevent some patients from developing a full PMS phenotype or sort of developing a milder version of it as well. So there are lots of different ways that genetic data can be sliced and diced to help make sense maybe of what's happening, with the phenotypes in the patients.
Absolutely.
Yeah. So we have concluded. It's 1 o'clock, or it's 2 o'clock Eastern time, and we need to wrap up our webinar. But I'm going to finish looking through these questions, Alex, and I'll send you, if there's anything else that we haven't covered, I'll send them to you, and maybe you can write a quick response as we've done in the past, and we can circulate the replies. And thank you so much, and best of luck to you. And we'll stay in close touch, right? As you're working on this, you have more information to share. We'll repeat these webinars. And yeah, that'd be perfect. Thank you, Al.
Okay. Thank you.
Bye-bye.
Bye.